Okay, I'm live. Hey, if anybody's on the other stream, it wouldn't go. So hopefully you guys will find me on the other one. If not, I'll have it posted and learn or Canvas, and you'll figure that out. All right, so live from Dominican Republic, Costanza. Check this out. There's in the mountains. I'm in the central mountains here of Dominican Republic doing some birding. Yeah, and herping, annuling too, right? Oh no, what happened here? Did I lose my stream? Oh, there it is. I got my binocs, got my bird book, and herping fuel for the night. This is a monster. It's even in Spanish if you look closely. I drank it after this thing right here. The Red Bull. All right. So today what we're going to go over is basically these cell-cell interactions, right? And if you think about it, animals and plants are these multicellular organisms. And being a multicellular organism means that you've got all these different cells. They differentiate into different kinds of cells by expressing different genes, and a lot of that is based on signaling. They're talking to each other. So that's something that makes a multicellular organism kind of unique, is their ability for all of these different cells to talk to each other and coordinate their interactions. Sorry about that. So we had these cell-cell interactions, and one of the interesting things about this cell communication, a lot of it takes place across the cell membranes. And you know that something like 50% of the mass of a cell membrane, 50% is phospholipids, the other 50% are proteins embedded in these membranes. And in fact, there's this estimate that 10, 15%, maybe even upwards of 20% of your genome is basically on membrane proteins that are doing all kinds of things, from receiving signals to various different types of transport. So a large chunk of your genome is making proteins that are in your membranes to receive signals and, of course, transport things as well. So with the cellular messaging, we want to know how they talk to each other, right? Because this is important, because if you can't talk to each other, you can't have multicellular organisms. It just won't work. So there's all kinds of interaction that happens. And in fact, that ability for cells to signal to each other, to send, chemical signals, that's part of the evolutionary steps and the origins of multicellular life. And to go from, single-celled organisms to a multicellular organism like an animal, of course, required many events. I mean, you had to, going way back, even before there were eukaryotes, you had to have the evolution of cellular respiration, then the evolution of eukaryogenesis, or endosymbiosis, which gave you, of course, eukaryotic cells. And then eukaryotic cells are the only cell type to have ever evolved multicellularity. And they did it probably about 3 times: plants, animals, and fungus. So at some point, cells began living in colonies, and they had to talk to each other. And interestingly, even though eukaryotic cells are the only multicellular organisms, there are bacterial films out there and bacterial colonies where very different bacteria do talk to each other, but we're not going to do much about that here. So understanding the evolution of cell signaling, I mean, we learned a lot from studying yeast. Yeah, baker's yeast, the same type of yeast you use to bake bread or brew beer. One's really fun. One tastes good too. So at any rate, what we learned from yeast is that these cells, they can talk to each other. There's this, cell communication, which is a crucial step. And here it starts. Yeast can actually reproduce sexually and asexually. But by reproducing sexually, they've got mating factors. So you've got these 2 different types of yeast. You've got the A type and you've got the alpha type. And the way they melt is they combine into a single diploid cell. So you get these haploid cells with these 2 different mating types. One will release the alpha factor, and the other one, the A, has a receptor for that alpha factor. And if they do that, then the cells will merge. And since the original cells are haploid, they'll merge into a diploid cell, which is really interesting that they know how to do that. So they're talking to each other. This is the origins of cell-cell communication. And then, of course, once they form a diploid cell, Those diploid cells can then reproduce asexually by making more diploid cells, or they can reproduce sexually by making haploid cells. And we'll learn about, or you'll learn in genetics about meiosis, where you undergo 2 rounds of cell division to go from a diploid cell to a haploid cell. Now, by now you guys know me. I love the mitochondria. It's like my favorite organelle, right? Because without the origins of mitochondria, without having mitochondria in your cells, you would never get a eukaryotic cell. So life would be basically limited to prokaryotes, these much smaller cells. But because you take this energy converting organelle, the mitochondria, you can pack it into a eukaryotic cell, and these cells can become larger and more complex. Whereas if you try to do that with a prokaryotic cell, because they're making all of their energy across their membrane, they actually run out of energy as they become larger. Whereas a eukaryotic cell, you can continually pack in more and more mitochondria, and you can develop a much larger and more complex cell. And it's also, of course, as you'll learn later, that the mitochondria also made it possible not only for the origins of eukaryotic cells, but also for the origins of multicellular life. Remember, there are no multicellular prokaryotes out there, and part of it is they're not able to form these complex systems. Now, we start talking about this cell-cell communication. It's really important that we look at these proteins on the surface and get an introduction to the various proteins we have there. So a cell, life, it is a system. And a system is the set of these, there's these interacting components that are defined by a boundary. And the boundary to life, the boundary to a cell, is a cell membrane. And of course, you know this as a plasma membrane or a phospholipid bilayer. Both are very descriptive of the structure and function of these membranes. Now, we often draw them quite simply, right? We just throw a bunch of phospholipids in there and then draw these squiggly things in there for proteins. But these cell surfaces are, like I said, they are incredibly complex, with hundreds, if not thousands, of different types of proteins embedded in these membranes, all doing different things, from doing transport. Remember, from transport, we've got all of the uniporters, symporters, antiporters, and we have ones for potassium ions, chloride ions, sodium ions. You get the idea, all the different permutations there. But when we look at the surface of a cell, we start looking at these membrane proteins. We have ones called integral membrane proteins, and they are embedded. They are permanently attached to the membrane. And transmembrane proteins, this is a type of integral membrane protein, and they go all the way through the membranes. And a lot of times, if you remember, the secondary structure of a protein is made up of the alpha helices or the beta pleated sheets. Well, alpha helices become very important for embedding these proteins into the hydrophobic interior of these membranes. All right. So that's why we learn about that secondary structure of those proteins, because it allows them to do that. So transmembrane proteins are all of our ion channels, right? They're all of our active transport, our co-transporters, like the antiporters, the symporters, uniporters. They're also used for facilitated diffusion, voltage gated ion channels, right? Sodium pumps for active transport. So lots and lots of different types of transmembrane proteins that we've already learned about. Well, we've also got other ones called integrins, and a lot of these are responsible for cell adhesion, so cells can stick together. And we also have, not only are these proteins here, but we can also export proteins outside of the cell and form a whole entire extracellular matrix. Okay, so that's pretty cool itself, that we have all of these proteins outside. So integral proteins, they're helping cells attach to this extracellular matrix here. And so very complex here. Okay, so integrins are another type of these transmembrane proteins, and they have a couple functions here. One is they link the extracellular matrix. This is all of this stuff outside of the membrane, and they help link it to the interior of the cell, to the cytoskeleton. Remember, the cytoskeleton has the microtubules, the keratin fibers, and the actin filaments, right? And a lot of it's going to be attached to like actin filaments, and that's what gives some of the cells their strength. Other types of integrins, and here's our cell-cell interaction. They can transmit signals from the outs, receive them on the outside of the cell, and transmit that signal through the membrane to the interior of the cell. Okay, so you're getting signals from the extracellular matrix of a cell to the ECM. Okay, so that's important. So the cell surface, we've got lots of these different proteins here. We also have proteins that basically are not attached permanently to the membrane, but are part of the membrane, right? And some of these are, they can be attached to those integral membrane proteins. So one thing that we're going to learn about in um, our proteins here are called G proteins. And they're called a G protein because they have an activated nucleotide attached to them. And that activated nucleotide, you know of one, ATP. So a nucleotide, adenine, guanine, cytosine, thymine, an activated nucleotide has 3 phosphate groups attached to it, hence adenosine triphosphate. A G protein is guanosine triphosphate, right? So it's another type of activated protein. You can also have another class of proteins called kinases. Anytime you see the word kinase, what a kinase does is involved with the transfer of phosphates from usually an activated nucleotide like ATP, right? There's also electron carriers. So in this chapter, we're of course jumping ahead to chapter 11. We're going to come back and do photosynthesis and then cellular respiration after we talk about the cell-cell interaction. But involved with both photosynthesis and cellular respiration, what they're doing is they're making ATP through a process called chemiosmosis. So we'll talk about that. But basically, to do that, you have to have this electron carriers. They're taking electrons through the membrane of the protein, right? I mean, through the membrane. And so there's a lot of these electron carriers that are also going through our membranes as well. So like I said, there's lots of stuff happening here. All right. So when you think about these cell surface proteins, I mean, we've got integral membrane proteins permanently attached. And these include the transmembrane proteins. They go through the membrane and they are abundant. So think uniporters, symporters, antiporters, ion channels. And remember, you've got voltage gated, ligand gated, mechanically gated, gap junctions. Gap junctions actually help stitch these cells together. We got G protein coupled receptors. A G coupled protein, I know that is a very complex thing to say, G protein coupled. So you get the G protein on the inside, it's a protein with a guanosine triphosphate attached to it. And the coupled receptor is there's a receptor on the outside of the surface that basically receives a signal, transmits it through the surface, through the cell membrane, and what it does is it activates that G protein, which then sets off a whole set of events inside the cell. And then we also have the integrins. These are transmembrane receptors that are additional to the G protein coupled receptors. And these help attach the cytoskeleton to the ECM and are very important in the signal transduction. And let's not forget these cytochrome complexes used in electron transport. So integral monotropic proteins permanently attached to the membrane, only one side. We're not going to go into that. There's also peripheral membrane proteins, and they're found kind of temporarily attached to the cell surface. They can attach to these integral membrane proteins and work for them. Okay, so that's about as far as I'm going to go right now with introduction to cell surface. There's a lot to remember there. And like I said, we're going to go through this chapter 11 in Freeman's book in pretty good detail. And it looks like I'm getting some cell service has gotten a little, internet service, it's a little bit splotchy, but we're going to stop here today and we're going to come back into a shorter lecture again tomorrow and start picking up with the extracellular matrix. All right, let me see. I'm going to see if anybody has any questions here for me. We're all good? All right. So the PowerPoints, really, I made those PowerPoints to go along very nicely with the book. So we'll go from there. Like I said, Well, yeah, sorry, I got a little distracted here. There's cool martins flying around out there. You can see the backdrop. You can see the reflection in the windows here. But basically, we're going to do this chapter 11, and then we're going to backtrack after we do cell signaling and some of this other stuff, and then we'll come back to photosynthesis and cell respiration. Okay. Yeah, so all 6 PowerPoints, yeah, we're gonna do them. I'm gonna go back over them again. So yeah, we'll need to go through them. We'll see how far we get by Wednesday. As you know, I always stop on Wednesday. I don't try to catch up. But if I've got them posted for this week, yeah, I would go over them. I would read the chapter, and let's go do that. And the question is, do I offer extra credit? No, we don't offer extra credit in any of our core courses. Now, sometimes like the last test was out of 104 points. Yeah, I'll redo those tests and make sure that they're, the test will only count 100, but if you made 104 or whatever, there's a little extra credit on that. But yeah, go through those PowerPoints, read through them, and I've got some extra videos I can post on that as well. And if you have any questions, be sure to go ahead and hit me up on that, and we'll pick up tomorrow, and we'll continue on with this. All right, any other questions? All right. We'll see you tomorrow.